Hello students, and welcome to lecture 11 on the, actually this is lecture 12, I apologize. This is a lecture on, in the previous lecture, lecture 11, we learned how to deliberately grow bacteria and other microbes in the laboratory in order to either study them or to identify them. In this lecture, we're simply going to talk about ways to kill bacteria and other microbes. And there's a difference between whether you're trying to kill bacteria and microbes on surfaces. For that, you generally use things called disinfectants and things like that, or whether you're trying to kill bacteria and microbes that are infecting somebody's body and making them sick. There's quite a difference, because in the former category, if you're just looking to disinfect surfaces, then you can use fairly harsh methods to kill the bacteria. But if you're trying to kill bacteria that are inside the body of a living human being, for instance, you have to use gentler and more specific methods, which will only damage the bacteria or the microbe while not damaging the human. So those are two very different assignments. Those are two very different objectives. So in this one, we're going to talk about using methods to kill microbes on surfaces, in the air, on instruments, on fomites, and so on. And so that's what this lecture will be about. Okay, so chapter 13 from the OpenStax textbook, we will talk about various methods to explain killing microbes on kitchen utensils, on kitchen surfaces, in hospital operating rooms and surgical instruments and so on. The idea being to reduce nosocomial infections and to generally sanitize things. And then chapter 14 will talk basically about antibiotics. And so antibiotics, of course, are special chemicals that target metabolic processes in bacteria, but leave the human alone, so that they will harm only bacteria and not the human being that they've invaded. So let's begin. Okay, first of all, what do you want to kill? If we're talking about killing things on surfaces, are you interested in killing the endospores? Because killing endospores is generally much more difficult than killing just vegetative cells. So endospores are not actually alive. They're sort of like a seed. You may remember from first year biology that a spore is like a seed, except that it contains a haploid genome for an organism that may be a diploid organism or something like that. So generally a spore is a container that contains a haploid genome, a single copy of a genome. If you look at a plant seed, what a plant seed actually is it's a plant embryo, which is diploid or even tetraploid or quadruploid or something like that. But it's a developing embryo that has been arrested in its development. But it's not haploid, it's diploid or triploid or tetraploid. Okay, so an endospore is not alive. It contains a bacterial genome. It's contained in a case that's usually made of protein, and that case is quite durable, so it's very difficult to actually kill the spore. Okay, now on the other hand, if you want to sterilize So if you're interested in sterilizing surfaces, how harsh are the treatments? You know, can people survive it? There are a lot of treatments that will kill bacteria that will also kill a human, so they're not very useful. In some cases, you're using physical methods like heat or radiation, which may actually damage what you're trying to sterilize. And in other cases, you're using poisons that selectively poison bacteria or other microbes. And you have to ask, will those poison human beings as well? So obviously there are a lot of a lot of disinfectants that will poison bacteria, but you don't want to put them in food in order to sanitize food, for example. Okay, so before we go on, we should discuss a few definitions. Right, so definitions for killing bacteria and other microbes. Right? And we're also going to, later on in this lecture, we're going to discuss methods for determining how effective our microbial killing techniques are. So that we have some standard assays, some various methods when you're trying to kill bacteria or other microbes using various methods. You want to have some standard way to measure how effective the killing method is. Right? We want to have some way to measure that. So we will discuss some of the standard methods that we use to measure how effective these killing tools are. Okay, now this lecture on how to kill bacteria and other microbes will be divided into two parts. The first part will be about physical killing methods, killing microbes and other bacteria and other microbes with heat, with cold, with radiation, with ultrafiltration. Ultrafiltration is forcing fluids through a filter that has pore sizes in the filter that are so small that the bacteria will actually remain on the other side of the filter, and the filtrate, when you filter a liquid through a filter, what comes through the filter is referred to as a filtrate. So the filtrate will be sterile because the pores were too small to let bacteria through. I should mention that there are no filters that have pores that are small enough to retain viruses, because viruses are in the neighborhood of 10 to 100 nanometers in size. So they just don't make any filters that have pore sizes that are so small that they will actually retain viruses, but there are lots of filters that will filter out bacteria and fungi. And then the second half of the lecture, we'll talk about chemical methods, basically poisoning bacteria, and how harsh are those chemicals, what sorts of surfaces can you use them with, and so on. Okay, so under the category of physical methods, we're going to do autoclaving, which is basically just killing bacteria with high pressure steam that's heated to beyond 100 degrees Celsius. We'll talk about boiling. By definition, when you boil water at normal atmospheric pressures, the temperature is 100 degrees, not higher. So if you want the temperature of boiling water to go higher, you have to increase the pressure, which is what happens in an autoclave, by the way. So that's why an autoclave is around 1.5 to 2 times the normal atmospheric pressure on the inside, and therefore that allows the water, the steam from boiling water, to be at a higher temperature, typically up at around 120 degrees instead of 100 degrees. Right. Now, if you boil things for a very short period of time, or if you just heat them up for a very short period of time, you can treat certain foods that way, like milk, and we call that pasteurization, after Louis Pasteur, the famous French microbiologist. Okay, obviously, if you set things on fire and burn them up, you'll destroy anything living. So there are certain things like glassware, or particularly metal, like a loop. We talked about streak plating in the last lecture. You sterilize the loop in between streaks by putting it into a flame, so you burn off any of the bacteria. And also you can cook things in an oven. If we're talking about cooking things in an oven, we're usually talking about glassware, bottles, graduated cylinders, things like that. You can kill things under certain conditions by freezing them. Now, we'll talk about this because it's a very subtle, nuanced subject. That there are sometimes you want to stop bacteria from growing without, let's say you have food and you put it in the refrigerator. A typical refrigerator is 4 degrees. The reason why you put it at 4 degrees is because metabolism stops at 4 degrees generally, but freezing doesn't happen until 0 degrees. Now, if you literally freeze something at a temperature below 0, if it contains water, the water will freeze into ice crystals. Ice crystals take up more room than liquid water. You may have learned that in the chemistry class. So the ice crystals take up more room than the liquid water. So if you freeze cells, the ice crystals will actually burst the cells. And that's why when you freeze meat or vegetables, when you thaw them back out again, they seem limp and they're not quite as firm anymore. And so there's a very different taste and texture to food that's been frozen versus food that has not been frozen. So you can take food and you can cool it down to 4 degrees, and if that food contains bacteria or fungi that are going to spoil the food, those bacteria and fungi will stop growing at 4 degrees, but they're still alive. So if you raise the temperature, if you take that food out of the refrigerator and it's out in a warmer environment, the bacteria will continue to divide for a while until you put it back in the refrigerator at 4 degrees. And if the pot or the container that you have the food in has been heated, if you put it back into the refrigerator, it will actually take quite a bit of time before it cools down to 4 degrees again. So that gives the bacteria and the fungus that are in the food a lot of time to continue dividing and growing. That's of importance if you're in the restaurant business, or if you're in the restaurant inspection business. If you're a health inspector who goes around inspecting restaurants, that's something that you need to know. Putting food in the refrigerator and taking it out to heat it up, and then putting a hot container of food back in the refrigerator, and that hot container of food will take a long time to go back down to 4 degrees. That's something that you need to know about if you're a restaurateur. That is actually, there's an official term for that. That's called temperature abuse, where you're trying to keep something cold in order to preserve it, but you keep taking it out of the refrigerator to heat it and then putting it back, taking it out, heating it, putting it back. And the periods when the food is cooling down again give the bacteria a lot of time to grow. Okay, then we'll also discuss another technique which is called desiccation. Desiccation means to dry out. You've seen that. If you've ever seen beef jerky, which they sell in the stores over the counter, they sell beef jerky in the store over the counter. It is beef that has been dried out. It's cooked beef that's been dried out. Now, if bacteria and fungi are going to, and other microbes are going to spoil the food, they're going to consume the food, it has to be relatively moist, because bacteria and fungi and other protists, for example, they need a certain amount of water to live. And so if they're going to live on a surface and eat the surface has to be moist. So if you dry out the surface to a certain extent, you will actually prevent things like bacteria and fungus from growing on it. Okay, I already mentioned ultrafiltration. And finally, this will probably surprise you. Radiation is a way of sterilizing and sanitizing food that is actually very effective and perfectly harmless. Other than the fact that in order to irradiate food, you have to dispose of radioactive isotopes, which is bad for the environment. But other than that, the technique is actually very useful at cleaning up and sanitizing food. It's perfectly harmless. And as you'll learn later on in this lecture, you're probably unaware of the fact that a lot of your food is already irradiated. For 50 years or so, they have been irradiating wheat and cereals in order to prevent insect eggs from hatching in it. For probably 30 years, they've been irradiating fruit and vegetables at the supermarkets in order to prevent them from spoiling from bacteria. So I will talk about radiation, and you may be surprised to hear that I'm actually a big advocate of irradiating food in order to preserve it. Okay, and then at the second half of the lecture, we'll talk about chemical methods, and each of these chemicals that's listed on the right here is a specific type of treatment, a specific type of bacterial poison, and that will poison bacteria and other things. So we'll talk about each of those chemical methods for sanitizing things and chemical methods for killing bacteria. Okay, now there's a difference between the term bactericidal versus bacteriostatic. So as I mentioned, if you freeze bacteria without protecting it in any way, you will probably kill it. So that would be a bactericidal method. So if you put bacteria at below 0, you will probably kill most of it. That's bactericidal. If you put it at 4 degrees in the refrigerator instead of the freezer, you will not kill it. You will simply prevent it from growing. And if you take it up to a higher temperature, if you take it out of the refrigerator and put it at a higher temperature, it will continue to grow. So the word static means to stand still. Right? So bacteriostatic techniques are techniques that simply stop the bacteria from growing, but stop bacteria, fungi, and other microbes from growing, but they don't actually destroy them. Okay, so you know the term homicide. Homicide means to kill humans, and the word homa in homicide stands for Homo sapiens. Right? So homicide means to kill humans. Bacteriocide means to kill bacteria. So bactericidal techniques are different than bacteriostatic techniques. So as we go through all of these techniques, try and imagine, does this actually kill bacteria and fungi, or does it simply stop them from growing, or does it simply inhibit their growth or slow down their growth? All right, now some of these definitions are legally binding depending on where you live. You can't claim that some technique will sterilize something when all it does is reduce the number of bacteria present, for example. So the word sterilization legally means to kill all of the bacteria, fungi, and so on. And in order to actually sterilize something, that takes a very harsh treatment indeed. It takes a very harsh treatment to kill everything. Pretty much the only thing that we have that can do that is the autoclave. So you heat things up under high pressure steam, and you hold it under high pressure steam for a long period of time, and you will successfully kill everything if you do that. And so sterilization generally means that you have to use an autoclave. The word disinfectant means that you kill the live cells on a surface, but not generally the endospores. The word sanitize technically means any of a number of weak measures that can be used to simply reduce the number of germs or bacteria or fungi that are on a surface. The word septic means that something is dirty with germs, and so the term aseptic refers to, the word aseptic means clean with an absence of germs. And the word antiseptic means measures that you take that kill germs, right? So we have a number of definitions here: bactericidal versus bacteriostatic. We have sterilization as opposed to disinfection, sanitization, and then the word septics. The words septic, aseptic, and antiseptic are all terms that you should be familiar with. Okay, let's go through some examples. The word sanitation means, the word sterilization means to kill absolutely everything. Generally, that includes things like hospital operating instruments. If you're going to do surgery on somebody, the instruments must be sterile. They absolutely must be sterile, otherwise you'll contaminate somebody and give them a nosocomial infection. And generally, the only machine that we have that can accomplish that is an autoclave. So this is an autoclave here on the right. It has a boiler down in the bottom part. Down in the bottom part here, there's an electric boiler. It's either electric or gas, natural gas. And it literally boils the water into hot steam, and it pumps the steam into the chamber here where the things that you're trying to sterilize are. And you have to close this door to withstand the high pressure. So this is kind of like a submarine hatch or something like that. It's a door that you have to crank it to close it, and it's meant to withstand high pressure. So operation of an autoclave is actually quite dangerous, and so not everybody is allowed to use an autoclave because it can be quite dangerous. You have to be trained how to use the autoclave first. But once you've been trained, it's actually quite simple to operate. Okay, now there's another term called commercial sterilization, which refers to, it's a very technical legal term which refers to cooking food like soup, for example, in a pressurized steam colander, and you have to cook the soup, for example, until you have done what's called 12 decimal reduction times for Clostridium botulinum endospores. Okay, so what that means is legally that means that if you have some food that's contaminated with Clostridium botulinum endospores, you have to cook it long enough until you have destroyed at a high enough temperature and a long enough period of time to destroy 12 decimal reduction times for Clostridium botulinum endospores. Okay, now 1 decimal reduction time, we'll talk about this again later, but 1 decimal reduction time means cooking something at a specific temperature for a long enough period of time to kill 90% of the microbes, 90%. Right, so 2 decimal reduction times would be enough to kill 99% of the things present. 3 decimal reduction times would be enough to kill 99.9% of the microbes present, right? And so on. So it means you're whatever microbes are there, you're reducing them by 90%. That's 1 decimal reduction time. 2 decimal reduction times is if you take whatever's left and you kill 90% of that. 3 decimal reductions is if you take 90% of that, to kill 90% of what's left of that, and so on. So commercial sterilization for things like soup is actually quite a rigorous, strict regulation to make sure that these foods cannot be contaminated with Clostridium botulinum, because that would kill people. That would give you food poisoning. All right, what about disinfection? Disinfection is spraying soap or other things around on food preparation surfaces. You're not actually sterilizing the surface. You're simply just reducing the number of microbes that are there. Using these techniques, you'll never succeed in reducing them all the way down to zero. There will always be some microbes left. The word asepsis is a term that's used when you kill some of the bacteria on living tissue. Now, what you see here is somebody here is preparing a person's foot for surgery, and you might have seen this on television. When they prepare skin tissue for surgery, the skin looks kind of yellow. The reason for that is because somebody has smeared a disinfectant called povidone all over the skin surface. Povidone is a mixture of iodine and alcohol, and it has the effect of killing some but not all of the bacteria that you would find on a skin surface. Right, so so antisepsis in this case is preparing a skin surface for surgery. You're going to cut into the skin with a scalpel or something like that, and you don't want to carry any of the Staphylococcus aureus or anything like that that's on the skin surface down into the wound that you've created when you cut open the tissue. Okay, so that's antisepsis. Degerming refers to antisepsis of a small surface. So when they rub you go for a flu vaccination, the nurse will tear open a package that has a little tissue in it that's saturated with ethanol, and the nurse will rub that ethanol onto a little surface where they're going to poke the needle. And so that's called degerming. And sadly, when I was little, I used to think that they did that to lessen the pain of the needle. Turns out they're not doing it for that reason at all. They're doing it because they want to avoid the hypodermic needle bringing any bacteria underneath your skin and therefore causing an infection. Okay. Sanitation, very weak methods where you apply soap and warm water and so on just to reduce a certain number of the bacteria. So when you go to a hotel and they put this piece of tape over the toilet that says sanitized for your protection, that is a legal term which is practically meaningless. It refers to some very weak methods using soap and so on. Putting things in the dishwasher will clean and reduce the number of microbes, but again, it will not eliminate them. So a regular dishwasher will sanitize things, but it will certainly not sterilize them. Okay, so within this, sterilization kills absolutely everything, and then commercial sterilization is a term using to reduce significantly the amount of microbes in food, canned food, for instance. Right, so then disinfection, antisepsis, degerming, and sanitization don't do anywhere near that much cleaning. Okay, you might ask yourself which of these things are germicidal versus germistatic, right? So sterilization and commercial sterilization are definitely germicidal. The other things, in some cases, they're mildly germicidal, but they're usually not germistatic. Okay, now before we go on, I should mention about proper hand washing, and this is not just for doctors and nurses, but anyone, especially now during the COVID-19 pandemic, rather. It's critical to do a very good job of washing your hands, which is a way to avoid spreading germs and so on. Okay, so these are the steps that you should learn about washing your hands. The first step is to wet your hands thoroughly with water. And then you apply the soap after that. So if you just put on the soap and rub it around, or you put on the soap and then add a little bit of water, it doesn't do nearly as good a job. So then you put on the soap, you lather your hands completely, and then you scrub your hands for at least 20 seconds. At least 20 seconds. And by the way, that in order to time that, you can sing in your mind, you can sing Happy Birthday twice. So you know that song, Happy Birthday to You. You sing that to yourself twice, and that's 20 seconds. Right? And then you rinse the water off of your hands again. And then you dry your hands with a clean paper towel, or a clean towel, for instance, or you can air dry them. Right? And in order to make sure that this regimen for cleaning your hands actually works, you cannot touch the faucets or the handles And you can't touch the doorknob on the way out of the washroom if there is a doorknob, right? Because if you do that, your hands are dirty again and you've just wasted all of your time. So that's one of the interesting things about hand washing. So you may have noticed, for instance, in Columbia College that uh, we don't have handles on the faucets. And that's a modern invention. We have a motion detector under the faucet to make sure that you don't have to turn the handles on or off, because then you would just be dirtying up your hands after you'd cleaned them. Secondly, not everyone who uses the washroom washes their hands. They're supposed to, but you can't guarantee that everyone does. And so if somebody with dirty hands grabs the door handle to get out of the washroom, then how are you supposed to get out without touching the same door handle and recontaminating your hands? Right, so you may notice again that at Columbia College the washrooms don't have doors. They have a kind of a corridor that you have to go around so that nobody can see inside. But the reason there's no door is so that you don't have to grab a handle. Now, if you find yourself in a washroom that has a door and a handle, what you should do is take a clean paper towel and use the clean paper towel to grip the handle on the way out. If there are no paper towels, then you can pull your sleeve down over your hand and grab the handle that way. But you should do everything you can to avoid touching the handle on the door. So here, this is the way they do it in operating theaters and so on. There's usually a faucet. The faucet is controlled with a foot pedal. There's some washrooms that do that the same way. So here's the proper method of actually washing your hands, making sure you wash the fingers and so on. So you want to avoid touching the handles on sinks, and so we've just explained there are several ways that you can avoid that. One of the ways, if there are faucets with handles, you can take a paper towel and use one side only of the paper towel to open and close the faucets. And then, of course, over here there's a door handle here, and notice that somebody has put the waste paper basket right next to the door because they expect that you'll open the door with a paper towel, and then you just throw the paper towel in the waste paper basket, which is right there. Right? And if you don't have a paper towel, you can just use your sleeve. Okay, so all of that, by the way, means that washing your hands is very important, but it's not the most effective way to avoid getting an infection, because um, as soon as you come out of the washroom, as soon as you wash your hands, as soon as you touch a doorknob, as soon as you touch a railing on a staircase, as soon as you touch a button for the elevator, your hands are dirty again. Right, so that's why it's important to wash your hands as often as possible. But if you're worried about getting infected by COVID-19 or some other virus that's spread by droplets or by mucus and so on, one of the important things is to avoid touching your face, especially touching your eyes, because that's usually the way you get infected. You touch a doorknob that has somebody else's mucus on it, and then you rub your eyes, and then you've been inoculated. All right, so now let's move on to the subject of how do we measure how effective each of these killing techniques are. And there are different sets of methods depending on whether you're using chemicals to poison microbes or whether you're using physical methods to kill microbes. Okay, but we do have a few specific assays that are used to determine the effectiveness of killing techniques. All right, so these two methods are for killing bacteria with high temperatures, killing microbes with high temperatures. Okay, so if you're killing microbes with a set temperature, and you're asking, okay, if I put them at this temperature, how long do I have to leave them there to kill 90% of them? That is referred to as a log decimal time, or a log decimal reduction time, or a d-value. Right? So a d-value, or a log decimal time, is the amount of time that you have to leave an object at a certain temperature in order to kill 90% of the microbes that are on that object. Right? So that's one thing. Right? You can test how effective this is by using something called a Bacillus subtilis spore strip test, which I'll talk about in a minute. Or you can so that's if you have a set temperature and you want to know, and you have a variable time period. What if you have a set time period and you want to know what temperature to use in order to kill 90% of the microbes? In that case, you know the time, you want to know the temperature, and that is referred to as the TDP, or the thermal death point. That is the temperature that you have to use in order to kill 90% of the microbes in 10 minutes. Okay, now if you're killing things with chemicals, there are three other tests that we have to worry about. One is you just take a certain number of microbes and then you mix them with that chemical, and then you measure what's known. You take a bunch of samples, you take several aliquots, and then you put the bacteria in with this much of the chemical, and then you take another sample and you put it in with twice that much chemical, and you take another one and you put it in with 3 times that much chemical, 4 times, 5 times. You leave them all for a certain period of time, and then you measure how many bacteria are still alive, and then you plot them on a graph. And that way you can figure out how much chemical you have to use in order to kill 50% of them. And that gives you a value called the LD50, the lethal dose 50. Now, unfortunately, that's a little bit unfortunate because that's the same term that we used to talk about when we were talking about pathology and epidemiology. The LD50 was also what we used when we were infecting guinea pigs with um, uh, infecting guinea pigs with bacteria or viruses, and then we'd say how much bacteria do you have to infect these guinea pigs with in order to kill half of them, and that would give you an LD50 as well, lethal dose 50. And there was another term that we used called the ID50, which was the infective dose. So how much bacteria do you have to expose these guinea pigs to before 50% of them will get infected? Right. So now we have another definition for LD50, and in this case, we're talking about the LD50 as the concentration of chemical that you have to use in order to kill 50% of the bacteria that are present. You'll see that there's another term that you can substitute for this, which is called the LC50, which stands for the lethal concentration 50%, and so that might help to clear up some confusion. So you can use that term as well. Okay, now there's another method called the disk diffusion test, which is also known as the Kirby-Bauer test, and we'll talk about that. Basically, what the Kirby-Bauer test is you simply take little tiny round disks of filter paper. Have you ever punched holes in a document with a document hole puncher? You know how you get these little round pieces of paper? If you take one of those hole punchers and instead of punching regular paper, you punch holes in filter paper, you end up getting these little round filter paper discs. Then what you do is you dip that filter paper disc in a chemical, and then you take a bacterial plate and you spread plate a certain type of bacteria all over the plate, right? So let's say you're testing the effectiveness of killing Staphylococcus aureus. You take an LB agar plate, you spread Staphylococcus aureus all over that plate, and then you put one of these filter paper discs down in the middle of it, and then you put it in the incubator, and then you take it out the next day, and the plate will be covered in Staph aureus, and you'll see where the Staph aureus grew, except you'll probably see a little clear zone, a clearance zone, around the disc. And you can measure how big that clearance zone is, and the bigger it is, the more effective that particular chemical was at either killing or inhibiting the bacteria. So that's called a Kirby-Bauer test. I'll show you what that looks like. And then finally, there's another test that is not used too often that's called the use dilution test. It's where you dip some metal rings into a chemical, and then you cover some metal rings with bacteria, and then you dip them into a chemical, and then you and then you take them out of the chemical, and then you put them into LB liquid medium, and then you look to see if any bacteria still grow. Right, so we'll discuss all of those in turn. Okay, so these methods are only for killing things with high temperatures. Okay, so as we already mentioned, the log decimal time, or the decimal reduction, 1 log decimal reduction, DRT, decimal reduction time, is how you know what the temperature that you're using is, 100 degrees or 120 degrees. How long do you have to leave the object that you're trying to sanitize or sterilize in that temperature in order to kill 90% of what's there? So that's called a DRT. And so in this case, you know the temperature, you don't know the time. In the other one, the thermal death point, you know the time, 10 minutes, and then how long do you have to leave the object at what temperature do you have to put the object at in order to kill 90% of them? Right, so this is where we we don't know, we know the temperature, we don't know the duration. And here we know the duration, we just don't know what temperature to use. Okay, so in order to determine the thermal death, in order to determine the thermal death point, or in order to determine, in order to determine the decimal reduction time, you would do a series of experiments like this, where you take a test tube full of bacteria and you put it at that temperature for 5 minutes, and then you take a separate one and you put it at that temperature for 10 minutes, and then take another tube with bacteria and put it at that temperature for 15 minutes, until you have a whole bunch of time points. And then you would figure out how many bacterial cells or other types of cells are still there, and how would you go about measuring that? Right? Well, we talked about three different ways to measure last time. We talked about densitometry, we talked about CFU per mil spread plating, and we talked about a hemocytometer. So let me ask you a question. If you were trying to determine this value, if you were trying to determine what the D value is at 120 degrees for Staphylococcus aureus, which of those methods would you use to count how many cells you have? Right? Think about that for a minute and be prepared to justify your answer. Okay, and my answer would be I would use the CFU per minute, because densitometry, the spectrophotometer, densitometry, and the hemocytometer, unless you use the special stain, they would tell you the total number of cells, but they would not tell you the number of viable cells, the number of live cells. So what we need to know is the number of live cells. So anyway, so you can see here that in this case, whatever they've done this experiment and they plotted what's called a death curve, and then they can figure out how much time you need to leave the object at that temperature in order to reduce the number of microbes that are there by 90%. And you simply take all of the time points and you plot a graph. Right, and once there, in some cases, this is another way that you would do it. You would plot the graph like this. Right, so you would plot death curves to determine how long you have to leave surgical instruments in the autoclave, for instance. All right, now I mentioned a commercial sterilization of soup, which is sometimes referred to as a botulinal cook. A botulinal cook. Right, so commercial sterilization is where you have to cook soup for 12 decimal reduction times relative to Clostridium botulinum endospores. Right, so that's a long time. That's actually quite a harsh treatment. Right, so how long would you have to, at what temperature and at what pressure and for how long would you have to cook soup in order to achieve 12 decimal reduction times for Clostridium botulinum endospores? Because that's the standard that the soup industry and other canned food industries are being held to, right? And so they have to be very careful about this. And that's why when you have prepared foods like soups and things, theoretically you can eat it without having to, you heat it up because you don't like to eat cold soup, but theoretically you could eat it straight out of the can without getting sick because it's been commercially sterilized, the commercial sterilization of 12 DRTs. Okay, so let me just ask you the question one more time. What would be the percentage of microbes left if you did 3 decimal reduction times? Right, so if you did 1, you would go from 100% bacteria, whatever percentage of, whatever number it is, you would have 100% of the bacteria live. If you cook them for 1 decimal reduction time, you would be down to 10%. And then if you cook them for 2 decimal reduction times, you would be down to 10% of 10%, which is 1%. Right? And then now you've got 1%. You cook them for 3 decimal reduction times. You've got 10% of 1%, which is 0.1%. Right? So after 3 decimal reduction times, you have reduced the number of either live microbes or viable Clostridium botulinum endospores down to 0.1% after just 3 decimal reduction times. Now imagine how few you would have if you went all the way to 12 decimal reduction times. Right? And that's what they have to do under commercial sterilization techniques. All right, now how do you measure? We already said, how do we said we've discussed these methods that we're using to kill the microbes. What specifically, if you're if you're trying to measure the effectiveness of an autoclave, for instance, let's say that you have an autoclave and you suspect that it's not working properly, you want to make sure that your autoclave is actually killing everything in there. And so there is a specific bacteria that you would use to test whether your autoclave is working or not. And it is a bacteria called Bacillus thermophilus. Bacillus thermophilus is a relative of Bacillus cereus and a relative of Clostridium botulinum, and it produces endospores, but it's perfectly harmless because it doesn't produce any toxins. Right? So Bacillus thermophilus, they sell Bacillus thermophilus endospores on things that are called spore strips. So they take a little tiny, basically a piece of tape, and they tape a whole bunch of they stick a whole bunch of Bacillus thermophilus endospores to that tape. And then what you do is you take that strip of tape with the spores on it and you put it into your autoclave, and then you sterilize it, theoretically. And then you drop it, once it comes out of the autoclave, you drop it into a flask full of LB medium, and you look to see if anything grows. And if nothing grows, then you can be assured that everything was killed in the autoclave and everything was sterilized. Right? If you drop the spore strip in and then put the flask in the incubator and it turns cloudy, it becomes turbid, that proves that it wasn't sterile and there's something wrong with your autoclave, and that happens fairly often. So if you work in a hospital or a research laboratory where they have an autoclave, you're required to do the Bacillus thermophilus spore strip test every month or something like that, and then you have to sign it. There's a sheet on the side of the autoclave that you have to sign that says that you did the spore test and it's working properly for that month. Right, so you do this treatment using the spores, you put it into liquid medium. If the liquid medium becomes turbid, it means that the spores were not killed. So this is a commercial spore strip that you would buy this little pouch, this package from a company that sells them, and you tear the pouch open, and inside you have about 100 of these little strips of tape. They look kind of like a bandage. You pull the top layer off of the tape and you've got the spores exposed, and then you put that into the autoclave, and then you take it out of the autoclave, and you can even autoclave it inside a flask of LB medium, and then theoretically nothing should grow because you sterilized both the spore strips and the LB medium at the same time. All right, so that is basically, so you use the spore strips to test your autoclave. And then you can also use the spore strips to test the effectiveness of your food preparation technique. So there's a company that makes soup. They take one of the colanders that they're making the soup in. Typically, a company like Campbell's Soup would have dozens of those pressure colanders that they're cooking the soup. They would take one of them and they would put a batch of soup in there, and they would put a spore strip in there with it. And then after they've done the cook, they would take the spore strip out, and then they'd put it in some LB medium and see if anything grows. If it does, then you know that soup colander is not working properly. You would throw the soup away that was in there with the spore strip, because you don't want people to be eating Bacillus thermophilus in their food, but it's just a way of testing the effectiveness of the soup colander that you're using. All right, now what about killing things with chemicals? Okay, so the lethal dose, or the lethal concentration 50, is the concentration of chemical that you need to kill 50% of the microbes present. You would do a series of those little experiments, each one of them, several test tubes, each one of them would have a little more of the chemical than the previous one, and then you plot death curves like that, right? Okay, now obviously if you're trying to sanitize things with chemicals, you wouldn't be satisfied with just killing 50% of them. Right? You would not be satisfied with killing just 50% of the microbes present. You'd want to kill 90% or 99%. But the only reason why we care about determining what the LC50 is because that allows us to compare the effectiveness of different killing chemicals. Right? So if the LC50 for chemical A is 100 millimolar, and the LC50 for chemical B is 5 millimolar, you tell me which one is a more effective killing chemical. Is it the one that has an LC of 100 millimolar or an LC of 5? obviously it would be the one that has the LC50 of 5, because you have to use much less of that chemical in order to kill 50% of the microbes. So don't get confused. We're not actually interested in killing 50% of the microbes. We're just finding out how much we need to use to kill 50% of the microbes in order to assess the effectiveness of that particular chemical. Okay, the disk diffusion method is it's also known as the Kirby-Bauer test. You take a filter paper that's saturated with a chemical, you put it onto a plate, and then you measure the clearance zone. And then the use dilution test, where you take bacteria that are on discs and you dip them into chemicals. All right, so lethal concentration 50, you plot death curves using increasing concentrations of the chemical in order to figure out how much you need to add in order to kill 50% of the bacteria that you started with. That would give you this number here, whatever that number is. In this case, that looks like somewhere in there. Somewhere in there. Okay, the Kirby-Bauer test. We take these little filter paper discs and we dip each one of them in a different chemical, and then we put them onto a plate where we have spread plated Staph aureus or E. coli over the entire plate, and then we look to see how big the clearance zone is. You can actually take a ruler. What you're actually supposed to do is measure how big the clearance zone is around the disc, and then you can compare. If one of them has a clearance, one chemical has a clearance zone of 3 millimeters, and another one has a clearance zone of 1 millimeter, which chemical is more effective at killing that bacteria? And the answer is the one with the larger clearance zone, right? So when we were talking about lethal concentration 50, the chemical with the smaller number is the more effective. If you're talking about the Kirby-Bauer test, the chemical that gives you the larger clearance zone is the one that's the more effective. Right. Okay, so you notice up here, chlorine, that's bleach. Right? So bleach is a very effective method of killing Staph aureus. It's a very effective method at killing E. coli. And it's a fairly effective method at killing Pseudomonas aeruginosa. Now, these other chemicals that are on these other discs, notice that they kill some bacteria but not others. They're very bacteria specific, and none of them are effective at killing Pseudomonas aeruginosa, because in fact, Pseudomonas aeruginosa actually likes quaternary ammonium compounds, quats, and we'll talk about that later when we get to the chemical section of this lecture. So the use dilution test, you dip some discs into bacteria and then you dry them out. So these discs, these metal rings are covered with bacteria, and then you dip them into a chemical, and then you take the disc that you dipped into that chemical, and then you put it into a flask of LB medium, and then you look to see if anything grows. And that's a sort of a pass-fail method of measuring whether a chemical works to kill microbes. All right, so you dip the disc into the disinfectant, right, and then you put it in, and then you take it out, and then you put it into some medium, and then you do a bunch of serial dilutions so that you can actually quantify, you can actually count the number of bacteria that survived. That's the use dilution test. All right, so measuring the effectiveness of killing with heat, we would use the log decimal time or the thermal death point, and we would do this with spore strips that contain Bacillus thermophilus. Okay, for chemical methods, we would use the lethal concentration or the lethal dose 50. We would determine what that is so we can compare the effectiveness between two different chemicals. We would use the Kirby-Bauer test and measure the clearance zone, and the chemical that gives you the larger clearance zone is more effective. Or we could actually quantify how many, we could again quantify the way we did with the LC50 using the use dilution test. All right, what physical methods are available for killing microbes? Okay, so the most obvious one is the temperature. So high temperatures will kill microbes, and so will low temperatures with certain caveats, with certain conditions. Now, others, filtration and desiccation, salt, these adding salt to things, these are actually bacteriostatic techniques, right? So if you take a piece of beef jerky and you dip it in, you put it in some water, whatever bacteria that were on it will start to grow again, and they will cause the beef to go bad. They will cause the beef to rot. If you take the filtration device, you filtered some liquid, that liquid is now sterile because the filter filtered out the bacteria, but you didn't actually kill the bacteria, you simply removed them from whatever liquid you were trying to sterilize. Okay, so let's look at measures dealing with temperature. In the old days, they used to boil surgical instruments for 10 minutes, right, in order to sterilize them. We've since learned that a number of things like hepatitis B can survive that from time to time, right? So you can, boiling is not considered to be sufficient for dental instruments or surgical instruments. You have to use an autoclave. An autoclave, you use hot steam at high pressure for at least 15 minutes, at least. And then there are various other settings that you can set the machine for, but generally an autoclave is killing things with 121 degrees Celsius steam. 30 psi is 1.5 atmospheric pressures. I believe it's like 1.5 to 2 times higher the regular atmospheric pressure. And then you have to leave it under those conditions for at least 15 minutes. You can kill things like metal loops for doing streak plating using the flame. And you can sterilize glassware by baking it in an oven at 170 degrees Celsius for 2 hours. For freezing, you have to freeze the bacteria. You have to freeze the object that you're talking about at minus 20 degrees generally. It has to be below zero. And this causes the formation of ice crystals, which cause bacteria and fungi to burst because the crystals are bigger than the liquid. Now, there's an interesting caveat here, which is that sometimes we want to preserve bacteria in the refrigerator, basically. It's not good enough to put them at 4 degrees because they kind of die. Eventually, they'll die at 4 degrees just because they've exceeded their lifespan. But if you want to freeze them in the freezer, which you often do in a lab. So let's say you have a laboratory that's studying Staphylococcus aureus, they would freeze it in the freezer, and then they would have samples of Staph aureus in the freezer. And what they would do is, in order to stop the crystals from bursting the cells, you have to mix the cells with something called a cryoprotectant. Cryo means cold temperatures, and then protectant, of course, means to protect from cold temperatures. So the cheapest cryoprotectant that you can get is glycerol. You can buy glycerol at the pharmacist over the counter. It's not a dangerous chemical. And so the glycerol, if you have a sample of bacteria, let's say you have a culture of bacteria, if you mix glycerol with the bacterial culture to a final concentration of 50%, the glycerol will get inside the cells and prevent ice crystals from forming. Right, so if you mix a bacterial culture with 50% glycerol, make it up to 50% glycerol, and then you freeze that at minus 20 degrees or even minus 70 or an even colder temperature, the bacteria will actually still be viable. They will not form ice crystals, and they will be viable for use so that you can thaw them out and use them for many years, decades into the future. Right. So in laboratories that really want to preserve, that are really serious about preserving their bacterial specimens, they mix them with either glycerol or another chemical called dimethyl sulfoxide, and they put them in liquid nitrogen. I don't remember offhand what the temperature for liquid nitrogen is, but I think it's like 200 degrees below zero or something like that. Liquid nitrogen is a liquid, of course, and so you have to put it into a large container called a carafe. Or a carboy. So you have which is basically a giant thermos bottle. And so you know that if you want to keep your drinks cold, you put them into a thermos that has a silvered glass on the inside. If you have a giant thermos bottle like that, you can put liquid nitrogen into it, and then you put in bacterial specimens inside tubes, and that's a way of preserving them. Okay, so you can use freezing to either kill things or preserve things depending on whether you've mixed them with a cryoprotectant or not. Remember that refrigeration at 4 degrees is germostatic, not germicidal. Okay, so these methods are meant to kill everything. The other two methods are bacterial or germicidal. All right, so as I said, boiling for 10 minutes is not always effective. That's why hospital instruments and dental instruments must be autoclaved. So this is an old-fashioned hospital boiling instrument. You plug it in, it's electrically heated, and the electric heat would boil the water. So that was a way of, these are still used in some parts of the world. If they can't afford an autoclave, they might still be using commercial boilers, but be aware that that doesn't kill everything. In particular, hepatitis C sometimes can survive this treatment. I told you about earlier in the course about the bedroom dentist in Burnaby, bedroom dentist of Burnaby. He was running a dentistry out of his own house in one of the bedrooms in his house, and he was charging his customers very low fees because he didn't have to worry about all the details that you have to worry about when you have a real dentist's office. So he didn't have a lot of the equipment that a real dentist would have, but that's not necessarily a good thing. Sometimes, in fact, government regulations, most of the time government regulations are put there to protect you, not to interfere with your business or whatever. So after this guy was discovered, he escaped to Toronto, but he was captured in Toronto, and the police went on television and made an announcement that all of his customers should be checked for HIV and hepatitis C because he did not have an autoclave. He was just boiling the instruments. Okay, now speaking of boiling and heating liquids, um, pasteurization is a treatment that they invented to sanitize milk. So before they started pasteurizing things, people would get brucellosis fever. They would catch Brucella abortus and other things from contaminated cow's milk. So the trick is, Louis Pasteur figured out if you boil the milk, you will sanitize it and you will kill most of those bacteria. But the trouble is that uh, you will destroy the taste of the milk by boiling it. In fact, you'll make the milk curdle. It'll coagulate. So he figured out through experimentation, he figured out empirically at what temperature can you get away with heating the milk and for how long without destroying the flavor. And so the original pasteurization technique, named after Louis Pasteur, called for heating the milk to 63 degrees Celsius for 30 minutes. Right, so that was the original pasteurization technique. We've since improved on that modification, which is to use something called high temperature short time pasteurization. HTST pasteurization refers to 72 degrees for 15 seconds. They've determined that if you heat milk to 72 degrees for 15 seconds, it's equally effective at sanitizing the milk as if you had heated it to 63 degrees for 30 minutes. The only difference is that 63 degrees for 30 minutes would have destroyed the taste of the milk, and it would taste kind of flat and burned tasting milk. But the high temperature short time treatment is an equivalent treatment. So this is another thing that you have to be aware of, another term that you have to be aware of in microbiology. When they talk about equivalent treatments, that usually means low temperature, a long time exposure to a low temperature is sometimes equally effective as a high temperature exposure for a short period of time. Right, so the original pasteurization was 63 degrees for 30 minutes until they invented the equivalent treatment of 72 degrees for 15 seconds. So there's Louis Pasteur. So when you hear the word pasteurization, we're referring to cows and heating up their milk. We're not referring to cows in the pasture, not referring to that at all. And this is in the middle here, you see a commercial pasteurization machine that does the high temperature short duration. It's a very expensive machine. Only large dairy processing plants can afford this. And basically it's constructed so that the milk flows through pipes, and then one part of the pipe is really hot, and then the next part of the pipe next to that is really cold. So you heat the milk up for only a few seconds, and then you cool it right back down as fast as you possibly can. And then that has the effect of not destroying the texture and the flavor of the milk. So autoclaving, of course, we have a boiler down in the bottom. It forces steam into the chamber for a long period of time. That actually kills everything. This is a desktop autoclave. The one that's on the far right is a hospital-sized autoclave. You can see it's probably 2 meters in height or taller. This little benchtop one is about the size of a bread box. You know, it's about 20 centimeters by 10 centimeters, something like that. And you plug it in, and this is ideal for dental instruments, right? So if you have a dentist office, this is really the only kind of autoclave that you need. But if you have a high turnover where there are lots and lots of if you work in a dental clinic where they have a high turnover of patients, where there are lots of people going to the clinic every day, and you have to sterilize dental instruments in a larger volume, you'd probably have a larger autoclave like the one on the right. This is a very cheap autoclave that's meant to be put onto the stove. And it has basically, it just heats up the water inside and keeps it at a certain pressure, and it has a safety valve which will open up and let some of the pressure out. We have one like this in the Columbia College biology lab for sterilizing our microbiology plates after the biology 110. This is cheaper, not as good, not as flexible. You can't do as many fancy things with this, but it does work to sterilize instruments. Okay, flaming the loop is obviously possible if you're doing streak plating. Right, so here we have somebody streak plating, somebody's flaming the loop on the left with a Bunsen burner. If you're working around flammable chemicals, you may not want that. You may not want an open flame. And so on the right, we have an electric flame, an electric sterilizer. So you simply stick the metal loop into that little hole, and then you leave it on while you're working. You can see there's an on-off switch down there at the bottom. So you leave it on, and then you just stick the loop in here for 10 seconds or something, and you've sterilized it. Right. So as I mentioned, I had a friend who set the whole bench on fire when she was trying to do streak plating, because the alcohol flames tend to burn very dim, and so you can't see that the glass rod is still going. So the same thing could happen sometimes with a loop. And so if you don't want to have an open flame around, you can use one of these electric loop sterilizers. Right, so here you have somebody, a technician who's streak plating a lot of plates, and she has a Bunsen burner, not an electric one. Okay, baking glassware in the oven. Here's a glassware oven. This is literally called a glassware oven. So obviously this is only for fomites. You can put glassware as well as metals in there. You can put metal things in there like needles and picks and scalpels. All right, now freezing. Okay, so here on the left we have a guy that's in the United States Army Germ Warfare Division. They do actually have in the United States Army they have a germ warfare unit, and he's got things like anthrax in that freezer. Now notice that the freezer is not a vertical refrigerator like we have at home. At home you open up the door, as opposed to having a chest freezer that you have to bend over to lift the door open. And the reason why a chest freezer is actually more efficient because cold air is more dense than warm air. So if you have cold air in the refrigerator, if you open up the door and it's a vertical refrigerator, the heat falls out. The cold air falls out because it's heavier, it's denser than the warm air. So as soon as you open the door to your refrigerator, the cold air will spill out basically onto the floor. Warm air will replace it, and then you have to shut the door and let the refrigerator cool down again. But if you have a chest freezer like this, you open the lid at the top and the cold air stays in the bottom. So this is actually much more efficient. So what the guy on the left is using is a -20 chest freezer. So he's storing bacteria at -20 degrees. On the right, we have a liquid nitrogen carboy, which is a great big huge thermos that this woman is putting. She's wearing thick gloves in case she splashes any of it on herself. You have to wear glasses, goggles, safety goggles in case you splash any of it in your face. And so she's putting bacteria into the liquid nitrogen. The liquid nitrogen will boil off as any liquid will, and so you have to replace it. You have to keep on filling this thing up continuously in order to keep the bacteria frozen. Okay, so here's our home refrigerator. Technically, the freezer is usually kept at -20 and the refrigerator is kept at 4 + 4. Whether or not it will stay that temperature depends on how crowded it is and how often you open and close the door. Right? So if you open the door all the time, it's more likely that the refrigerator, instead of being 4 degrees, it will be up at 6 or 10 degrees. And so there'll be lots of opportunity for the bacteria to keep growing at 10 degrees. The freezer, if you keep it closed all the time, it won't warm up, but the more stuff you put in the freezer, the harder your refrigerator has to work in order to cool it off. So by having an overcrowded freezer, you'll make the refrigerator work too hard, and you'll end up having to buy a new refrigerator sooner than you otherwise would have. And so that's something to bear in mind if you're going into the restaurant business or something like that. Try to keep the freezer um, uncluttered, because it's a lot of work for the refrigerator to cool, the Freon compressor to cool off that much stuff. Okay, so I mentioned temperature abuse at restaurants. This is where you make a pot of soup, and then you serve some of the soup to people, and then you put it in the refrigerator. So when you boil the soup, it was at some point, presumably it was up at 100 degrees. You put a 100-degree pot into the refrigerator, and if it's a deep pot, if it's a large, deep pot, it will take a long time for it to cool down to 4 degrees. Right? So during that time, the bacteria can continue to grow. Right? So if you keep doing that, you cook it, you put it in the fridge, takes a long time to cool off. You take it out, you heat it up again, serve some of it, put it back, takes a long time to cool off. If you keep doing that over and over again, you've given the bacteria lots and lots of time to grow. Right? Whatever bacteria survive the heating process will have lots of opportunity to grow. So that's called temperature abuse. Now you can avoid this if you're a health inspector for restaurants, or if you're a restaurant owner and you're working in a restaurant. You can avoid this by storing these soups and things in a shallower pan or a shallower pot. And I'll show you how this works. Okay, now if you have, let's say you have steamed rice, for instance, if you have it in a deep pot, 15 centimeters, this is how long it takes to cool off from 80 degrees down to 10 degrees. Right? It takes 8 hours. If you have a shallow pot that's only 5 centimeters, it only takes 2 hours to cool off. Right? So if you store things in shallower containers, if you're heating the vessel up and cooling it off, heating it up, cooling it off by putting it back in the refrigerator, it will cool off much faster and give contaminating microbes less time to grow if you have a shallower pot. Right? So bear that in mind. And notice over here on the right, notice that this experiment was done with steamed rice containing Bacillus cereus endospores. They were wondering how much, and Bacillus cereus in general, because remember, Bacillus cereus is a common contaminant of rice, and so you don't want to be taking rice, steaming rice, and then taking it in and out of the refrigerator, re-steaming it, heating it, cooling it off, heating it, cooling it off, because it will actually make the customers sick. Because Bacillus cereus, and as I said, the trick of trying to take steamed rice and then saying, well, maybe it's contaminated, but I'll clean it up by killing the bacteria that are in there by heating it up, by turning it into fried rice. Yes, indeed, if you take the steamed rice from yesterday and you fry it today, you will kill the vegetative cells, but the Bacillus cereus toxin, it produces 2 toxins. And one of those toxins is heat labile, which means that it's destroyed by heat. And the other one is heat stable, which means it's not destroyed by heat. So Bacillus cereus growing in the rice will produce 2 toxins, one of which you cannot kill by heating it. One of which you cannot kill by heating it. Right? So it doesn't help to turn steamed rice from day 1 into fried rice from day 2. All right, filtration. So you can get filters. You can get filters that are used in the lab that have various sizes of pores, including 0.22 microns and 0.45 microns. Right? So how big is a bacterial cell? We discussed the fact that they're generally around 1 micron. Right? So 1 micrometer. So that means that if you have a 0.45 micron filter, you should be able, in theory, to sterilize a liquid, because the microbes, if you filter the liquid through a 0.45 micron filter, the bacteria are too big to fit through the pores. Right? Okay, but there are some bacteria that are really thin, like spirochetes and spirilla. And so in that case, you might want to go to a more expensive filter that is 0.22 microns instead of 0.45. So the 0.45 micron filters are slightly more expensive than 0.22 micron filters, but both of those are used commonly in labs to sterilize liquids that cannot be heated. So if you find yourself working in a microbiology lab one day and they ask you to prepare a solution of vitamins to feed to microbes or something like that. You cannot heat vitamins because you'll destroy them. The only thing you can do is mix them up and you'll and then filter them. So there are many cases where you work in a microbiology lab or a biology lab where you have to have sterilized solutions which you cannot sterilize by heating them. And so the best solution is to filter them. Right? So that's the technical term for that is ultrafiltration. Right? Now, desiccation means to dry something out so that it's so dry that bacteria and fungus can't live on it. Lyophilization is a combination of drying things out and freezing them. You freeze dry them, right? So you put some food, for instance, into a vacuum that's cooled down to a low temperature, or you have something and you take some soup and you freeze it, and then you suck all the water out of it while it's being frozen, and that's actually the most common way that they make powdered soup mixes through lyophilization. So they simultaneously freeze and evaporate the water out of the soup, and then you end up with just powder. And the powder is dry enough that bacteria and fungus will not grow in it. So then you put that into pouches, and then you open up the pouch and you pour it into a bowl and you add boiling water and it will come back to life again, basically. Next, osmotic pressure. Specifically, in this case, we're talking about just making a salty solution. Remember that in first-year biology, we talked about hypertonic solutions versus hypotonic solutions and isotonic solutions. Well, what you do in this case is you make a hypertonic solution, and whatever you put into that solution, the water will be sucked out of it into the hypertonic solution. And if you succeed in sucking enough water out of it, that means that you will stop the bacteria from being able to grow, because bacteria and fungi need a lot of water to grow. Typically, bacteria needs more water than fungus. And so just bear in mind that fungi will always grow on things with less water than bacteria. So it's relatively easy to remove enough water from certain things to prevent bacteria from growing, but eventually fungus will grow on those things if there's still a certain amount of water in it, because fungi can grow on a fairly low amount of water. The fungi are at a disadvantage if it's a competition between bacteria and fungi. Bacteria usually win because they grow faster, but they also require more water. So fungi, they grow more slowly, but they require less water. So if you're talking about an environment that's slightly dried out, then the fungi have the advantage. And then finally, radiation. There's a difference between ionizing radiation and non-ionizing radiation. Non-ionizing radiation usually means ultraviolet light. You can generate ultraviolet light. You can get ultraviolet light from the sun. You can also get what's called a germ lamp, which is an ultraviolet light. And that will kill many bacteria and fungi, versus like X-ray radiation, a radiation source that generates X-rays. X-rays actually have the ability to shatter the DNA double helix, so that you'll actually break and smash and destroy the genome of these microbes that are contaminating the food. Filtration. Right, so you pour something through a filter that has extremely small pores, and you can successfully sterilize things through ultrafiltration. This is technically called ultrafiltration. Here in the middle, we have what's known as a syringe filter, right? So this is a syringe, right? So let's say you only have to make up 5 milliliters of a liquid that you need to sterilize. You wouldn't want to buy a whole container like this. These containers are made out of plastic, by the way, and they're disposable. So you can only use this filter once. It's a fairly expensive way to sterilize something. But you wouldn't want to waste one of these whole containers if you're only going to sterilize 10 milliliters or 5 milliliters of this liquid. So then you suck the liquid up into a syringe. The syringe doesn't necessarily have to be sterile. And then you screw a syringe filter onto the end of the syringe, and you push the liquid through the syringe filter. Okay, so that's a syringe filter. On the right, we have a larger filter for filtering about a liter worth of liquid. Okay, now you've already become familiar with a HEPA filter when we talked about laboratory techniques. So remember that the HEPA filter is a filter that filters the air inside a laminar flow hood. A HEPA filter stands for high efficiency particulate air filter. And so this technician is working inside what's known as a laminar flow hood, and the air inside that hood that cabinet it's called a biological safety cabinet, which is sometimes called a hood, HOOD. Biological safety cabinet is a much better term, actually. So the biological safety cabinet, the air that's inside it is being filtered and by the HEPA filter. So the HEPA filter is a way of HEPA filter is not perfectly sterilizing. It doesn't sterilize things completely. In good working condition, the HEPA filter will remove about 70% of any fungi or bacteria or fungal spores that are in the air, but not 100%, but around 70%. Okay, now let's see what happens if we can preserve foods and other things by removing water so that there's no longer enough water in that food for bacteria and fungi to grow. Okay, so you've seen, I mentioned this, beef jerky is where you take a cooked piece of beef and then you dry it out in a machine called a desiccator. You should also understand that many of you are vegetarian. Congratulations. I think it's very good to be vegetarian, but some of you eat meat, and so you know what I'm talking about. You know that in Western civilization, we have this obsession with smoked meat, smoked flavors. Originally, people got exposed to the taste of smoky meat because they had things called smokehouses. You would go out and you would kill animals and you would skin the animal and then you would hang the animal in the smoke hut and then you would put a whole bunch of leaves and hickory smoke was what they used, and they would expose the meat to smoke. And they didn't know why this worked, but that would preserve the meat and it wouldn't go bad for a long period of time. So smoking meat was originally a method of preserving it, and then people decided that they actually liked the flavor of it like that. But in fact, originally it was a way of preserving the meat, and the way it worked was that the smoke desiccated the meat. It actually sucked the liquid out of the meat because you had a whole bunch of the smoke actually attaching itself to the surface of the meat and sucking the water out of the meat that way. So that was how smoking meat was a way of preserving the meat. Now they simply use a desiccation machine that sucks the water out with low pressure in order to make beef jerky like this. This is a lyophilization machine. It has a receptacle up here at the top where you put liquid nitrogen and that cools off the sample, and then there's a vacuum pump over here that sucks the air out. So you're simultaneously, if you put a piece of food in there, you are simultaneously sucking the water out and also freezing it at the same time. After it's been lyophilized, you can take away the cold and you just end up with a powder. And that powder, so that's the main way that they make powdered soups and things like that. Interestingly, if you're a microbiology scientist and you're interested in sending bacteria to your colleague through the mail, that's usually the way they do it. You take bacteria, you lyophilize it down into a powder, and then you put the powder in a little tiny tube, like an Eppendorf tube or something, and then you send it through the mail to your colleague at another laboratory. Your colleague opens it up and adds water, and then you've got viable bacteria again. All right, this method we've known about for centuries. When people started sailing the oceans and they were trying to take meat and vegetables with them, they would pack them in salty water. All right, so here you have a barrel full of pork that the English are carrying around in a ship or something, and the pork is sitting in very salty water. That has the effect of drying out the meat and preventing bacteria from growing in it, right? So salt is a preservative. So if you ever come across a health food store that has a sign out front that says we don't use any preservatives, you can go in and you can ask them if they have salt in any of their foods, and if they say yes, then you can say, well, that sign is not accurate then, is it? Because salt is the most widely used preservative, and it is able to preserve the food because it sucks enough of the moisture out of the food in order to prevent bacteria and fungi from being able to eat it. We've known this about pickles, right? So you take cucumbers and you put them into brine solution. Brine is just salty water, and then they kind of shrink because the fluid is being sucked out of them into the salty water. All right. Many of you may not realize that a lot of the food that you eat, the vegetables and like spinach, the fruits when you go to the store and you buy strawberries and raspberries and blackberries, they have been preserved through ionizing radiation, so X-rays. Right, so you expose the food to very powerful X-rays. That has the effect of shattering and smashing the DNA genome of the bacteria and fungi that are on those foods, and then those things are dead, so they can't eat the food. Right, so and it doesn't do anything to the food because the food is already dead. Right, so it doesn't matter whether you smash the DNA of the spinach. It doesn't matter because the spinach was already dead. The same thing is true for meat or anything like that. Right, so now in case you're worried, the food does not become radioactive. Right, the food does not become radioactive if you expose it to radiation. Unless you touch it to the radioactive source and then you get some of the radioactive source on the food, then of course that would be bad. But other than that, the food never touches the radioactive source. The radiation is simply used to sterilize the food, and ionizing radiation comes very close to actually sterilizing food, which means that and it doesn't destroy the texture of it, right? So if you cook food, you'll kill most of the bacteria, but you'll destroy the leaves because they'll go limp and soft, whereas radiation doesn't do that. So radiation is actually a very effective way to sterilize food, and therefore it lasts longer. And I think it's a good thing to have more food in the world because there are people who don't have enough to eat, and so you can give them. If the more food there is in the world, the more likely it is, the lower the price will be, and the more likely it is that people that don't have enough food will get some. Furthermore, it takes land to produce food, so you do farming, right? So the less, if the food doesn't go rotten fast, food rots and then you have to throw it away and then you have to buy more. And so therefore the farmers have to produce more. But if the food lasts longer without going bad, then you don't have to throw it away and the farmers don't have to produce as much. If the farmers don't have to produce as much, then you don't have to use as much farmland to produce the food. You can use the farmland for other things. So I think having more food in the world is a good thing, and this is a way to increase the amount of food that's in the world. Unfortunately, people are kind of squeamish about irradiating food, and so my view is that if companies irradiate food because they think it's useful, and if you get government scientists to agree, hopefully the scientists have not been pressured by the politicians the way they often are, but scientists are often pressured by politicians and they shouldn't be. That should be a serious crime. But if you get scientists who have not been pressured by politicians, they work for a university and they have tenure and so they can't be fired for making an unpopular discovery the way government scientists can. If the company thinks it's good to preserve food this way, and if scientists agree that it's not harmful and it's nothing wrong with it, then the companies that want to do this should explain that to the public. They should spend money on advertising to explain to the public why irradiating food is not dangerous and why it's good to irradiate food, because if you sterilize food by irradiating it, then you don't have to put in a lot of harmful chemicals, which many companies do now. Right? So that's what companies who want to irradiate their food should be spending their money on. Instead, what they spent their money on was bribing politicians, which is technically called lobbying. When companies give money to politicians and say, okay, we'll give you this money to help you get elected, you can't put it in your pocket. The politician can't take the money and put it in their pocket. That would be a bribe, an absolute bribe. But what they can do is companies say, well, we'll give money to your election campaign, and then if you're elected, then you do what we say. And if you don't do what we say or what we advise you, then next time around we'll give the money to your opponent and see if they'll comply. Right? So that's not called bribery or intimidation or blackmail, but that's what it is. And so the companies that want to irradiate food, instead of spending their money educating the public about why irradiating food is not bad, they spent their money instead bribing politicians. Frankly, they probably spent less money because it's easier to bribe politicians than it is to explain things to the public. But that's what they did, and they successfully bribed the politicians into letting them say the words cold pasteurization on the package instead of irradiation. Right, so whenever you see a package of food that says that it's been cold pasteurized, that means that it's been irradiated. right? And they feel that the public doesn't know what that means. Many of you are probably hearing that for the first time, and you didn't. If you'd seen it before, you wouldn't know what it means. But that is what it means, right? So it means that the food has been irradiated. Okay, so these are all hazard symbols. The one here, this one here, is a radioactivity. It denotes a radioactive source. Right? The one in the middle is a biohazard warning, means that there's something biologically dangerous about it. And then over here is a radiation source warning as well. Which one do you think they put on the packages of irradiated foods? Right? It's none of these. It's this one. That looks very eco-friendly, doesn't it? That's a very eco-friendly looking sign. And that is the symbol for cold pasteurization, which indicates that the food has been irradiated. So you see this irradiated spinach has the cold pasteurization symbol down there. If you go to the store and you buy fresh fruits, they have probably been cold pasteurized. They've been irradiated in order to preserve them. If you don't, particularly raspberries, raspberries last about 2 days before they go bad. That's not even enough time to put them on the supermarket shelf. So you can check to see whether your raspberries have been cold pasteurized or not. My guess is that if you put them in your refrigerator and they last for longer than 3 days or something, they've probably been cold pasteurized. When I was a boy, we used to have cereals that my parents kept in a cupboard in the kitchen. And if you kept the cereals around for too long, you would have these little brown beetles that came out of the cereals. And also, my mother used to bake things with flour, and so she had a canister of flour under this cupboard where she did the cooking and food preparation. And if you left the flour in that can too long, these beetles would come out. These are called flour beetles, and they are found all over the wheat. And when they grind the wheat into flour, many of the eggs of these beetles remain in the wheat, and then those eggs would hatch not only out of flour but out of prepared cooked cereals. Right? So this was a problem until in the 1960s, the various governments around the world gave these companies permission to irradiate the wheat. And so ever since, we haven't had flour beetles appearing out of the cereal boxes and things because the flour, all of the flour that's used to make cereal or bread or just if you're just buying flour, all of it has been irradiated. This is a map of all the countries in the world that allow cold pasteurization or irradiation. You can see Canada is one of them, obviously. This is the process of cold pasteurization. So this guy is putting these food parcels onto this conveyor belt, and you can see the radiation symbol in the middle here. So beyond that curtain, as soon as the food goes in, gets over onto the other side of that curtain, it's exposed to an X-ray machine basically that irradiates the food. Okay, now pharmaceuticals, generally they are kept sterile by irradiation. So when you take pills, they're in a soft plastic bottle that would never stand being autoclaved. So the bottle has to be sterile and the pills have to be sterile. So both the bottle and the pills were sterilized using irradiation. These hypodermic needles in the middle, notice that the hypodermic, you're aware that hypodermic needles are made out of soft plastic, and they could never stand being autoclaved, and so they are sterilized by irradiation. So when you have catheter tubes and IV tubes and hypodermic needles, all of these medical supplies that are made out of soft plastic, they have been irradiated in order to sterilize them. The needles you can't irradiate because unlike soft plastic, if you expose metal to radiation, the metal will become radioactive as well. So the syringes have been, the plastic syringes have been irradiated, the metal needles have been autoclaved. You probably know that if you go to the hospital, sometimes you need to get blood. If you've had surgery and you lost blood as a result of the surgery, they have to transfuse you with blood. The blood has been irradiated in order to kill any bacteria or any living cells that are in there. That includes white blood cells. Right, so the white blood cells in the pouch of blood that they're putting into you are technically dead because they've been irradiated. All right, what about non-ionizing radiation? I mentioned to you the fact that the ancient Romans used to pump all of their water through fountains. After a while, they made those fountains to be aesthetically pleasing. They're beautiful. But originally, they were there to clean up the water. The Romans had no idea what bacteria are, but they just knew that if you pump the water up into the sunshine and into the air, it was less likely to make you sick than if you didn't. If you just had it flowing underground in pipes and it flowed through the pipes, but it never got exposed to the fresh air and the sunlight, it would probably make you sick. And we now know that that's because most of the, or many of the pathogenic bacteria that would make you sick are number one, they are usually obligate anaerobes, and so if you pump them up into the air, that will kill them. And secondly, the ultraviolet radiation generally damages the DNA of the bacteria. Now, it doesn't shatter the chromosomes the way ionizing radiation does, so it doesn't smash the chromosomes into pieces, but it does cause mutations. It does cause mutations. Now, mutations are not a big deal, not necessarily a big deal for multicellular organisms like humans, because we have 2 copies of every gene, and we have multiple cells, right? So if you mutate one copy, we've got another one. You mutate one copy of a gene, we've got another one. If you actually succeed in killing one cell, we've got billions more, so it doesn't matter. The worst thing that UV radiation can do to us is to cause a mutation to a cell control gene, to a proto-oncogene, and therefore cause cancer. But and so you can get cancer from being out in the sun where the UV light is. But generally, other than that, we don't have to worry about that. Bacteria do, because they're haploid and they're single-celled organisms. So if you cause a mutation to a haploid organism, they don't have a spare copy of that gene, and you know, they don't have extra cells. Right? So that's why ultraviolet radiation is generally much worse to bacteria and fungi than it is to humans, for instance. So here we have the biological safety cabinet again. And notice that there's a blue light in there. That is called a germ light. And so when you're finished for the day working in this laminar flow hood, you turn it off, and then before you leave the lab, you switch on the germ lamp to kill any bacteria that are still hanging around inside the biological safety cabinet. So that's called a germ lamp. All right, let's move on now to chemical methods for killing microbes. Most of these techniques were discovered purely by accident, and the optimum or ideal dosage to use for killing bacteria of different types was determined empirically by experimentation. So these are mainly bacterial and microbial poisons, essentially. Right. So these microbial poisons fall into the following groups. Phenol, some of you that have taken chemistry have been exposed to phenol. You know, phenol is a very harsh chemical. And there's a related chemical called bisphenols. And some of those bisphenol chemicals go under brand names like triclosan. Right, so triclosan is found in many antimicrobial soaps. Okay, there's another class of chemicals called biguanides rather. And example, chlorhexidine, that's another brand name. The halogens like iodine and other things will kill certain bacteria, as will alcohols, aldehydes, and peroxides. We'll talk about those. Metal ions like silver ions, copper ions, and zinc ions will kill bacteria as well, and we'll talk about those. And then finally, there's a complicated class of chemicals called the quaternary ammonium compounds, which are based on the ammonium ion, therefore they're related to bleach. They're called quats. They kill bacteria as well. And then there are a couple of other specific chemicals that we don't really have any idea how they work, but we've been putting them into our food for centuries, literally for centuries. And so I'll talk to you about those as well. Let's start out with the phenols and the bisphenols. Okay, so the fact that phenol could kill bacteria was discovered purely by accident by a surgeon named Joseph Lister in the 19th century. And he discovered that you could sterilize surfaces and things, or you could sanitize surfaces and things by putting phenol onto them. And then he started up this tradition of doing surgery on patients while one of his assistants was spraying phenol through the air with a mister. And those of you that have taken chemistry, if you've ever been exposed to phenol, you know that it's a very harsh, irritating, caustic chemical. And it would be very irritating to your skin, very irritating to your eyes. And so somebody discovered that if you simply take two phenols and put them together into a bisphenol, then it's not nearly as irritating and not nearly as caustic as just phenol. So these days we don't really use phenols as a sanitizing agent, but we do use bisphenols. Okay, so now the phenols are very harsh. Bisphenol derivatives are less harsh. So you'll often find bisphenol chemicals in antimicrobial soaps and mouthwashes. But be warned that Pseudomonas aeruginosa actually likes it, so it won't kill them, right? So you should never be sterilizing hospital surfaces and hospital rooms and hospital toilets and sinks with anything that has the brand name triclosan in it, for instance, because that you're actually encouraging the growth of Pseudomonas aeruginosa, and Pseudomonas aeruginosa is a common wound infectant. Right, so here we have Lister trying to do surgery while his assistant over here has a mister machine, an atomizer, and he's spraying this phenol all over the place, and all of the assistants have a nasty look on their face because it's very irritating. Somebody discovered that if you make a chemical compound from a phenol called a bisphenol, it's not nearly as bad. Right, so there's the phenol. Right, if you make any of these bisphenols, they're much less irritating to the skin and eyes, but they do succeed in killing bacteria, most types. Okay, so triclosan is a brand name for one of the bisphenols. It's this one here, right? And if you look on a lot of these soaps and toothpastes and mouthwashes that claim to be antimicrobial soaps, you'll see that they have triclosan, right? But I don't advise using them because number one, soap kills bacteria anyway. So why do you need to add another chemical to the soap? Soapy water is an extremely good killer of bacteria because the soap will destroy the bacterial phospholipid bilayer. Right, so you don't need to add chemicals. This is one of those things that industry has pushed on us that we don't need, and in fact it might even be counterproductive. So you don't need to put triclosan into soap. In fact, you're making things worse because it actually encourages the growth of Pseudomonas aeruginosa. Okay, so notice down here Pseudomonas aeruginosa, right? So there's a quaternary ammonium compound down there, and there's an O-phenol, which is a bisphenol compound over there on the right, and you can see that the O-phenol does not even phase the Pseudomonas aeruginosa. The Pseudomonas aeruginosa on the Kirby-Bauer test is growing right up to the boundary of the filter paper disc. Next, the biguanides were discovered by accident again. They are frequently put into mouthwashes and toothpastes. An example would be chlorhexidine. Chlorhexidine is a biguanide that you'll find in a lot of these mouthwashes and things. Again, Pseudomonas aeruginosa actually likes it. Okay, the halogens. These are the halogens on the periodic table: fluoride, chloride, bromide, iodide. I don't know what AT stands for. Maybe some of you do. But for reasons which we don't quite understand, these things tend to interfere with bacterial and fungal physiology, probably because they substitute for prosthetic groups in the enzymes. They displace prosthetic groups from the enzymes. Okay, so they were discovered by accident. The fact that they kill bacteria was discovered by accident. You can use some of the halogens. Probably the best halogen to use is iodine. You can use it to purify, or not to purify, but to sanitize camping water. You can put iodine inside alcohol to make povidone, and you make a compound called povidone iodine, which is used to sterilize skin surfaces before surgery. And furthermore, if you put iodine into a tincture, you need to know what the word tincture means. Tincture is a mixture of something with a lot of alcohol. Right, so we already know that alcohols kill bacteria, and we're going to discuss that again in a minute. But if you make up 50% isopropanol and then you add something like iodine to it, you've made a tincture. And the tincture has synergistic effects. In other words, if you take 50% isopropanol or 70% ethanol, and then you mix in 1% iodine or 2% iodine, it kills much more effectively than either one of those things would alone. And furthermore, it kills more effectively than if you had twice as much alcohol or twice as much iodine by itself. So the two things work together synergistically so that it's better than either of them would be alone, or even better than either one of them would be if you had twice as much of it. Okay, so now, so that's iodine, one of the halogens. And in addition, chlorine, you notice that they put chlorine into swimming pools and they chlorinate drinking water to sanitize it, right? And probably what's happening in the case of chlorine is that the chlorine is denaturing proteins, including enzymes. Denaturing proteins, of course, means that you unfold them so they no longer have their 3D shape. The three-dimensional shape of an enzyme is necessary for its biological activity. So if you unfold or denature an enzyme, you'll kill whatever organism is using that enzyme. Phenol probably does the same thing. Right? Phenol probably does the same thing. Now, we live in an earthquake zone. Vancouver is in an earthquake zone, in case you didn't know that. Occasionally, we have had some little earthquakes in the past. I hope not, but we might someday have a big earthquake. I hope not. But typically what happens during an earthquake is that the ground water pipes will break, and so the groundwater may be contaminated with sewage. The water that comes to your house from the pipes may be contaminated by sewage. So you need an emergency method to sanitize the water, the drinking water, after an earthquake or some disaster like that. And so it turns out that if you put 2 drops of bleach, which are perchloride ions, into a liter of water and shake it up and leave it for 30 minutes, you can drink that water, and it will have killed most of the coliforms that are contaminating the water. It will have killed most of the coliforms that are contaminating the water. Right. Okay, so starting with the iodine. This is iodine put on skin surfaces. Depending on how old you are, your mother might have had an iodine bottle in the family medicine cabinet. And so if you were playing outside when you were a child and you fell on your knee and you skinned your knee and you got a cut, then your mother would have taken this iodine and put it on the cut, and it would sting and it would make you cry and scream and holler. And in fact, what the purpose of the iodine was to sterilize the wound so it wouldn't get infected. I used to think that it was supposed to stop the wound from hurting, but it certainly doesn't do that. It hurts more if you put the iodine on. And then iodine has been incorporated into things like povidone iodine ointment, which is a 1% iodine with other things mixed in with it. Chlorine. Okay, so you know that they pump chlorine gas into swimming pools because it's filled with people that have dirty ears and dirty noses and dirty mouths, dirty genitals, right? Dirty rectums. All of these horrible human body parts are put together into this soup, right? And so it's not swimming pool is not a sanitary place, so it's more sanitary if you put chlorine gas into it. Furthermore, you know that that the city drinking water in most cities is sanitized with chlorine gas. So the water plant in your city, in Vancouver, for instance, we get the water from Capilano Lake. It comes into Burnaby, and there's a sanitation ponds in Burnaby where they mix chlorine gas with the water before they send it out to the city to drink. So here we have a water treatment plant, a commercial water treatment plant, where the water is coming in and chlorine gas is being added to it. These green pipes contain the chlorine gas. So this is one of the tanks out over at Burnaby, where the water treatment plant is pumping the chlorine gas through the water and stirring it to kind of mix it, and then it gets sent down sent through the water pipes for us to drink. And as I said, if you take a 1 liter bottle of pop, 1 liter bottle of soda pop, for instance, and you drink it, you wash out the bottle. And then if you have water, if you keep water in it, everyone should keep an emergency preparedness kit in case we have an earthquake in Vancouver. So you should have a battery operated radio, but ideally a hand crank radio, a dynamo radio that you crank with a little crank to generate the electricity. And you can buy them over the internet through eBay and whatnot. They sell these emergency radios that have a crank that it's a combination of emergency radio and flashlight, and the flashlight is powered by the crank. And then you should keep some emergency food rations like canned foods and things like that, and peanut butter is very good. And then you need to keep an emergency water supply, so you keep some water, and then you also should keep a jar of bleach, a bottle of bleach, in order to, if your emergency water supply runs out and you have to start drinking the water out of the pipes, then you have a way of sanitizing the water. You just fill up one of these pop bottles, you have a bunch of these empty pop bottles. You fill one of them up, you put in 2 drops of bleach, leave it for 30 minutes, and then you can drink it, and it has eliminated most of the coliforms. Okay, alcohols, aldehydes, and peroxides. Okay, so again, these were discovered by accident, but probably alcohols have the effect of denaturing proteins and denaturing enzymes, which thus kills the bacteria that they were in. Right, so they are microcidal. They do actually kill. You have to use 70% ethanol or 50% isopropanol. If you use 95% ethanol, that will not kill the bacteria. That will actually just dehydrate them, and then if you add water, they'll come back to life. So be aware of the fact that more alcohol is not necessarily better. It has to be 70% ethanol. If you use more, like 80 or 90% ethanol, it will not kill the bacteria. And if you use 50% ethanol, it will not kill the bacteria. Similarly, isopropanol, commonly known as a rubbing alcohol, you need 50% isopropanol. If you have 75% isopropanol, you may not kill the bacteria. Or if you have 30% isopropanol, that may not kill the bacteria either. And I mentioned the fact that alcohols are synergistic with other things like iodine if you make them into a tincture. So 1% povidone iodine and 70% ethanol is a very effective tincture. There's something you can buy called Zephyran, which is benzene, a halogen compound, plus 40% bacteria. It kills 40%. Sorry, the Zephyrin kills 40% of the bacteria by itself, but if you mix it together with alcohol as a tincture, the two of them combined kill 85% of the bacteria. So I just mentioned that to point out that that's an example of synergy. Okay, rubbing alcohol is the common name for isopropyl alcohol. The reason I mention this is because many pharmacies will sell you isopropyl alcohol, but they won't sell you ethanol, because ethanol, they're worried that somebody's going to drink it as an alcoholic beverage, which I can't imagine anybody doing that, but it's more expensive than booze. Maybe they're worried. I don't know. I don't know what they're worried about. But for regulatory reasons, most pharmacies will not sell you ethanol, but they will sell you isopropanol. All right, so aldehydes, again, they're very strong denaturation agents, and so they'll kill enzymes. Now, formaldehyde is a very strong preservative. Glutaraldehyde is less strong and less irritating, but slightly less effective as well. But either one of these things will preserve things by killing bacteria and fungi. So you may have noticed in the biology lab at Columbia that we have preserved hearts and organs and kidneys and things like that. They are preserved in 50%. They're preserved in 10% formaldehyde. It's usually 50% glycerol, 10% formaldehyde is what they use to preserve biological specimens in the lab. So also, when people die, sometimes they get embalmed. So embalming fluid is where they basically remove the blood from the body and they pump formaldehyde in as a substitute. And so all the body tissues become saturated with formaldehyde, which has the effect of killing the bacteria, which would otherwise decompose the body. Okay, now glutaraldehyde is less irritating a chemical, but it's also less effective. So they tend not to use glutaraldehyde as an embalming fluid, but you can put small amounts of glutaraldehyde into a fish tank, into an aquarium, in order to kill any bacteria that are in the aquarium. So they do sell fish tank aquarium sanitizer solutions that contain glutaraldehyde. Right, so here are some preserved organs that you would see in the Columbia College Biology Lab. They are sitting in 10%, 50% glycerol, 10% formaldehyde. Here's a fish tank solution that contains a little bit of glutaraldehyde. You put 2 drops of that into your fish tank and it keeps the bacteria away. And viewing bodies at a funeral, the corpse of the person has probably been had formaldehyde. The blood is pumped out and then formaldehyde is pumped into all the blood vessels in order to preserve the body. That's what embalming is. It's not a very pleasant subject, but that's what they do at funeral homes. Okay, hydrogen peroxide is an oxidizing agent which cross-links proteins and therefore inactivates enzymes. It's less harsh, it doesn't sting. So you might well think, well, if I cut myself, why do I have to put iodine on it or alcohol if that stings? If you put hydrogen peroxide onto your wound, it doesn't sting as much. but that's not recommended because hydrogen peroxide will actually interfere with the healing process. So it is not recommended as it will sanitize things, but it's not recommended as something to sanitize a wound before you put a bandage on it. Okay, so not only is it hydrogen peroxide is an oxidizing agent, it's a free radical, and one of the things that an oxidizing agent will do is actually take the color out of things. It will take the pigment out of things. So you can buy hydrogen peroxide over the counter at the drugstore. It comes as a 3% solution, as you can see. People use it to bleach their hair, right? So it takes the color out of hair, and people use it to whiten their teeth. It also has the effect of killing some bacteria. All right, metal ions. Again, the mechanism is unknown, but these things will kill some bacteria probably by substituting for prosthetic groups in the enzymes of the bacteria. So most enzymes in bacteria and fungi have iron as or magnesium as a cofactor, as a prosthetic group. If you yank out the iron and substitute a copper ion, or if you pull out the metal, the iron ion, and you put in a silver ion instead, probably the enzyme will not work anymore. So metal ions have the effect of killing bacteria. Right, so now one application for this was that to this day, when a newborn baby is born, they will sometimes bathe the baby's eyes with silver nitrate, because it's a silver ion, will kill bacteria, in order to prevent the baby from getting gonorrheal ophthalmia neonatorum. Neonate means baby, newborn baby. Neonatorum means dead baby. Right? So gonorrheal ophthalmia neonatorum means gonorrhea that got into a baby's eyes and killed it through the eyes, ophthalmia. Right. So if the mother had gonorrhea, maybe if the mother has undiagnosed gonorrhea, it's easy to cure gonorrhea with antibiotics, easy. But if you don't know that you have gonorrhea, then there's a possibility the baby will get gonorrhea on the way out of the birth canal, on the way out of the vagina. So it was common practice for many years. I don't know if they still do it. I believe they do, but I'm not sure. It was common practice for many years to take a newborn baby and rinse the baby's eyes with a silver nitrate eye wash to prevent gonorrheal ophthalmia neonatorum. Also, there's some very expensive bandages that have silver impregnated into them, and the silver will prevent bacteria from growing on the wound as it's trying to heal. Zinc chloride is put into a number of mouthwashes. It's actually effective, unlike triclosan in mouthwash. We used to use mercury, but we no longer do because mercury is too toxic. Right? Now, mercury was the original treatment for syphilis, right? Okay, so as many of you know, I like to incorporate a little bit of history into microbiology class. So any of you that are into neoclassical studies, the neoclassical art movement, where the 19th century painters were painting ancient Greek gods from Greek mythology. So this is a neoclassical painting showing Venus on the left, Mercury on the right, and Cupid in the middle. Right? And there was a saying for gentlemen. You know, they're not really gentlemen if they were doing this, but the wealthy gentlemen would go and hire prostitutes, and the doctors would advise against that. And they had a saying that said, "One night with Venus will lead to 6 weeks with Mercury." Right? And over here, what that means is over here we have a brass syringe that the doctor would fill up with mercury and then insert into the man's penis and fill it full of mercury, which would be excruciatingly painful. The syphilis was not pleasant either, so it's hard to, it's a little bit hard to understand which was worse, the syphilis or the treatment for syphilis. But then that led to the idea of the Mad Hatter. You might have heard of a Mad Hatter, like in the Alice in Wonderland fable. Mad Hatter was a man whose penis had been ruined by syringes and mercury, and he had to use a catheter to urinate. Used to keep the catheter in the hat, okay, but that's why he wore the tall hat. But what about the madness? the madness came from the mercury, because mercury would have a lot of bad effects on the body. It would cause your teeth and hair to fall out, and it would also cause you to go mad. So that's where the term Mad Hatter came from a so-called gentleman who had who'd lost his mind as a result of syphilis and syphilis treatments, and that's where the idea of one night with Venus and 6 weeks with Mercury came from. And so Mercury did kill the syphilis because metal ions do that, but it's a very toxic substance, and so we don't use it anymore. Here's a Kirby Bauer test, essentially, where they put a dime, a nickel, and a little silver charm onto a plate, and the charm has been moved aside. So you can see that there are slight, small clearance zones around the copper penny and the nickel that the dime is made of, and no bacteria were able to grow underneath this silver charm bracelet charm. And so that's just a demonstration that metal ions do, in fact, have an inhibitory properties for bacteria. Okay, quaternary ammonium compounds, also known as quats, they are broad spectrum bactericidal, means that they will kill all types of bacteria, and we'll talk about broad versus narrow spectrum antibiotics in the next lecture. Benzalkonium chloride is a common quaternary ammonium compound. It goes by the brand name Zephyrin or Cepacol. So you will see Zephyrin or Cepacol mouthwashes, so-called antiseptic mouthwashes. Finally, let's talk about miscellaneous food preservatives, right? So these are chemicals that you add to food to discourage the growth of microbes, usually fungi. So most of these chemicals are, for the most part, fungistatic. They slow down fungus from growing. You put them into the food, which means that we who eat the food consume these chemicals as well. But they're put there as a preservative to slow down the growth of the fungus. So, example, sulfur dioxide has been added to wine for centuries to discourage fungal contamination of wine. Sodium benzoate has been added for centuries to cheese to slow down fungal spoilage of cheese. And calcium propionate has been added to bread for probably 100 years to slow down the spoilage of the bread. So the bread becomes, it's viable and eatable. It takes longer for the fungus to grow on the bread. Right? So those are standard food preservatives. They're not particularly good for us, but we consume them as well. There's a solution to that. That's to irradiate the food instead of putting those chemicals in it. All right, finally, this really should have been in the previous lecture, but it was included in this chapter of the textbook. I don't know why, but I'll tell you about it now. So let's just conclude our topic of killing microbes outside the body, and let's talk about techniques for dealing with how do you work with dangerous microbes in the laboratory. So in the last lecture, which is where this part should have really been, but just pretend that this is on the last lecture material. Right? So how do you deal with hazardous microbes in the laboratory? Well, the microbiologists have developed things that are called biosafety levels. We have biosafety level 1, 2, 3, and 4. And these are certain precautions and certain types of personal protective equipment, or PPE, that you have to use if you want to defend yourself and protect yourself inside working inside a laboratory where you work with certain types of microbes. So let's go through the different biosafety levels. You are responsible for knowing this for the course as well. Okay, so biological safety level 1, BSL1, is for a laboratory where you're working with common microbes. They're relatively harmless. Basically, all you have to do is wear gloves, a lab coat, and goggles. That's standard PPE, standard personal protective equipment. When you're done for the day, you wash your hands before you leave. You wash your hands before you have lunch. Right? That's a BSL1 lab. Most labs in the world are classified as BSL1. BSL2 is slightly more cautious. This is where you would grow bacteria like S. staph aureus. That's an opportunistic pathogen. You would work in a safety cabinet with a HEPA filter. BSL3 is more dangerous. typically you would work with something like Mycobacterium tuberculosis in there. In this case, you have to work in a special room that has a special negative air pressure, which means that if you have a room, some of the bacteria, if they're airborne, if the bacteria is released into the air, is that room airtight or not? The odds are that if it's a standard room, it's not going to be airtight. But there's a way around that. You don't want the bacteria to leak out of the room into the environment. If you have negative air pressure inside the lab, that means yes, it's not airtight and air will leak, but if air is going to leak, it's going to leak into the lab instead of out of the lab. So typically a BSL-3 lab is a special lab where you put on special protective equipment, you put on a hairnet, a special disposable lab coat, gloves, a mask, and goggles, and the lab itself has a negative air pressure so that air is never under any circumstances is air leaking from the lab to the outside, but air from the outside is leaking into the lab, and that air, before it gets to go out again, it's filtered through a HEPA filter before it goes out through an exhaust pipe in the roof. Okay, so that's a BSL-3 lab. Then in a BSL-4 lab, this is for the most dangerous things that you can imagine, like the Ebola virus. And this is where not only do you live in a lab that looks like a BSL-3 lab, but you work in a lab that looks like a BSL-3 lab, but you have to wear a space suit, essentially. You wear a biohazardous material suit, or also known as a hazmat suit, hazardous material suit, that has a positive pressure. So you're wearing this suit like a diver's suit, like a deep sea diver's suit, that has positive pressure on the inside so that the air is not going to leak from the laboratory into your suit if you have any leaks. If you have any leaks, the air is going to leak from your suit out into the lab, but not the other way around. Okay, so that's a biological safety level 4. Canada has many biological safety level 3 labs. BC only has one. It's at the BC Centre for Disease Control. And so BC Centre for Disease Control has a biological safety lab 3, which is where they analyze most of the tuberculosis samples. So if somebody's coughing and the doctor takes a sample of sputum, they send it to the BC CDC and they test it in that lab to see if it's tuberculosis. The only BSL-4 lab in Canada is in Winnipeg. So there's the Canadian Virology Research Laboratory is located in Winnipeg. That's the only place in Canada where they have BSL-4 containment. So when you go into a lab, you'll see this sticker on the door. This tells you what the different levels mean, and it will also tell you which level that particular lab is classified as. So then you know how careful you have to be when you're working in that lab. Here's a standard BSL-1 lab. So you see the guy is wearing gloves and goggles and a lab coat. He's working with bacteria, usually not dangerous bacteria. Everybody works with E. coli in their lab for various purposes. Okay, BSL-2, they're working in a glove box that has a HEPA filter above the glove box to sanitize the air. Here's a biological 3 lab like the one they have at the BC CDC. The one that they have at the BC CDC, there's a nice woman who works in there. She's a microbiology technician. She spends her whole life, not her whole life, but her whole day in that lab because it's a lot of trouble to get in and out of the lab. So she takes her lunch in with her and she eats it in a special room where she doesn't have to come back out of the lab to eat lunch. But so she spends the whole 8-hour day in that lab. And what it is you have a special airlock, right? Because remember the air pressure inside the lab is lower than outside. So there's one room that has a door to get into the room and then a door to get out of that room and into the lab. It has 2 doors. Right? And that is so the idea is that you never have the 2 doors open at once because that would allow the air pressure to equalize both in and outside the lab. So you go into this little airlock room, you go in through the outside door, you shut it, then you go in through the inside door, then you change out of your clothes and you put on special head to foot surgical gowns basically. And then you put on a shower cap for your head, you put on a mask, you can't be in there without a mask, you put on special gloves, and then you do your work in the lab all day. And people can put specimens in through a little doorway that has 2 doors. But when things go out of the lab, nothing is allowed to come out of a BSL-3 lab or a BSL-4 lab that hasn't been autoclaved. So over here in the corner, we have an autoclave that has two doors. One door faces the inside of the lab and the other door faces the outside. Again, the two doors can never be open at once. But the idea is that if the technician is going to send something out of the lab, she's finished working with it for the day, she opens the inner door, puts it in there, turns on the autoclave, and then she comes out. When she comes out for the day, she takes it out and throws it in the garbage. Now, in order to get out, you have to take a shower, right? So you have to, to get in, you can be dirty from the street, but when you come out, you have to take a shower before you're allowed to leave. So you come in for the day, you go into the airlock, you go into a changing room, you change into special clothes, then you do your work all day, then you come back out to the changing room, you throw away those clothes, you take a shower, then you put your street clothes back on, and then you leave. So it's a lot of trouble to go in and out, but this is an extra safety precaution to make sure that these pathogenic microbes don't come out. So here's a typical BSL-3 lab like you would find at the BCCDC. Okay, this is a BSL-4 lab, which is similar, except that this time the same safety precautions are applicable. You have to go in through an airlock. You have to shower to get out. You have to take a shower before you go out. But in this case, you're wearing a positive air pressure spacesuit, basically, so that there's negative pressure in the lab so that air will seep into the lab instead of out. And then the air is filtered through a HEPA filter before it goes out. And then you're wearing a positive pressure suit so that air from the lab will not leak into your suit if there's a leak. Okay, you might have seen this movie. This is from a movie called Outbreak, starring Dustin Hoffman. Interesting movie. If you ever get a chance to see it, I'd advise you to watch it because you see what life is like inside a BSL-4 lab, and it's kind of a good movie. There's another movie with Matt Damon that's come out called Contagion, which is another good movie. It shows a lot of people working inside a BSL-4 lab for a highly contagious, dangerous virus. So those movies are more interesting to people like us because we know what all these things are for. All right, so those are the biosafety levels. All right, so we talked about different biosafety levels for different types of bacteria. We talked in different types of laboratories that work with different types of microbes. We talked about terminology, bactericidal versus bacteriostatic. We discussed methods, physical methods for killing things, as well as methods for judging how well they're killing things, assessment methods to quantify quantitatively how well a certain bactericidal method works. These are the measuring techniques. We talked about the different chemical measuring techniques as well. And finally, if you feel like it, just go home and take a look in your medicine cabinet and look and see how many of those cleaners that you have contain triclosan or other things. Okay, there's some exercises at the end of this slideshow which I'll let you go through on your own, including some things where I show you different machines, that do different things, and you have to tell me what they do. Right, so I ask you what all of these things are. And I'll see you at the next lecture. Thank you very much for your attention. I'll see you at the next lecture on killing microbes inside the body with antibiotics.